Oh, thank you. Good afternoon to all of you. Thank you very much. I will present today a case study regarding the growth of cyanobacteria in a reservoir where I work and a synergy of this growth of cyanobacteria with climatic changes that we detected. So the idea is to give you as an example of a possible synergy between climatic changes, contamination, intrusion of a invasive species of cyanobacteria that occurred in the reservoir that I studied since 1971, as an example of what can be a very overall and general problem in Brazil, especially in the tropical and subtropical areas. And I'll finish with some information on this overall problem of invasive species of cyanobacteria, climatic changes, and water temperatures changes. Well, the reservoir I study is Lobo Broa Reservoir. The official name is Carlos Botelho Reservoir. It's a small reservoir. It has only 7 kilometers length, 1 kilometer width. Shallow reservoir built up in 1936 for hydroelectric power plants, as many other reservoirs, small reservoirs in Brazil. And we chose this reservoir in 1971 to work in the watershed and in the reservoir as a model for ecological, limnological research and the application of science to the management of the reservoir. So this is an overview of the reservoir. The reservoir has two main tributaries that are with growth of macrophytes that control the nutrient input to the reservoir. As I say, the reservoir is shallow. The average depth is 3 meters. This is a picture of the reservoir. This was the center of the University of São Paulo that I started there to help it to found and was director for 10 years. It was a center, it is a center for training people and research in water research, hydraulics and water quality and limnology. We have many courses and many of presents here went to this reservoir for lectures, courses, and seminars. Another view of the reservoir, and this is the upstream, the area with macrophytes at the entrance of the two tributaries. And then here an overview of the landscape, the general landscape, and the tributaries. Soils are mainly arenite, the yellow soil, mainly arenite, acidic soil, with some basaltic intrusions like these basaltic intrusions here, but the soil is poor, and we have hydromorphic soils along the rivers and the wetlands. Okay, and this is a study we did in one of the tributaries related to the soil uses and the water quality of the small watershed of one of the tributaries. Well, the climate has a big influence on the reservoir. This is an average of 45 years of measurements. We have in the reservoir a first-class climatological station that was set up in 1971. So this is an average of the functioning of the rainfall, solar radiation, and wind. The two main forcing functions that influence the reservoir are rainfall during the summer and wind during the winter. That that circulates the water in the reservoir. The water is well oxygenated, and as a consequence, this reservoir is a polymitic reservoir is a reservoir that does not stratify. And here only some information about the wind in different periods. Another effect on the reservoir, we started with this reservoir, but then we discovered that was a general picture for the southern part of Brazil. The reservoir suffers the influence of cold fronts coming from the Antarctic, and each time the cold front passes over the reservoir and over the many reservoirs that we have in the south and southeast of Brazil, we have mixing, and you can see here the effect of the cold front after the passage of the cold front, the wind after the passage of a cold front. So this was one more mixing effect in the reservoir. This is a picture of the reservoir before the cold front and a picture of the reservoir in the same place during the passage of a cold front to see the mixing effect in the reservoir. Okay, and this is, we have a permanent station that produces data in real time. So we have here temperature and other variables, but you can see the connection between the passage of cold fronts and the water temperature, the oxygen distribution, and you can see the cold front and the air temperature, how it falls after the passage of the cold front. So this is one more effect. So this was the picture until May last year. 45 years of information that were collected, and we knew very well how the reservoir was working and what the forcing functions on the reservoir. And the concentration of chlorophyll was in average around not more than 10, 20 micrograms per liter, and the phytoplankton was consisting especially of diatoms, chlorophycia, and there were very small percentage of cyanobacteria in this reservoir. This was the picture during the summer, and this is was the picture during the winter. You can see the distribution. This was taken with a fluoroprobe that distinguishes between chlorophycia, cyanobacteria, bacillariophyta, diatoms, and so on. But you can see above the vertical distribution, the percentage of the phytoplankton groups, and during summer and winter, and practically no cyanobacteria, very little cyanobacteria. Well, with the recent changes that were a consequence of the global changes, the dry period in São Paulo was in the southeast of Brazil, was very intense. You can see the drop in precipitation during 2013, 2012, 2013, 2014, and then 2015. So we could see here a drop in precipitation. We had much less precipitation, especially during the summer, and precipitation was very important because diluted the nutrients, washed silica for the reservoir, promoted the growth of diatoms. With this dry period and no precipitation, and besides that, another problem that was the increase of temperature. You can't see very well here, but during last July last year, the surface temperature was 2 centigrades above the 45 years average. That was a very important sign of changes in the reservoir. We have a permanent system of measurements here. We have a multi-sensor that gives real-time information from the reservoir, and you can see this temperature here, that you have peaks of higher temperature, especially occurred during the winter. And then after that, we start to detect changes in the phytoplankton composition of the reservoir. For that, we use it. This is not this reservoir. I'm just showing that because we use a technique, a satellite image of phycocyanin. We use this satellite image to detect the distribution of cyanobacteria in the reservoir. But this also brings another worry, that is, this is Guarapiranga Reservoir that furnishes water for 5 million people in São Paulo metropolitan region. And you can see the distribution of cyanobacteria here through this phycocyanin through a satellite image. You can see that all the reservoir is full of cyanobacteria, especially microcystis that are very producing microcystin, and is dangerous to human health. Just to show the technique. The technique applied to our reservoir showed then in this period, that is depicted here for July last year. You can see the distribution of cyanobacteria in the reservoir. And when we look at the species present, it was Cylindrospermopsis raciborski. Cylindrospermopsis raciborski is a cyanobacteria that grows in deep waters of African lakes. It is now spreading all over the world. We have cases of infestation of this cyanobacteria in Australia that produced cases of hepatitis. This cyanobacteria can produce toxins like saxitoxins. It is associated with not only with bacteria, but also with viruses. So it's a real danger. It can adjust its depth according to the solar radiation. It has very flexible genetic conditions to adapt to different situations. So it's a real danger. You can see here more the distribution of the cyanobacteria during July last year. And here you can see that in January, still last year, we have very few infestation. These are satellite images. And you could see that during some periods, we had total infestation of cyanobacteria in the reservoir. In June, May, June, October, we had to shut down the reservoir for recreation because of danger of problems of human health to fisheries. The reservoirs in São Paulo state and the interior of Brazil, they are very important for recreation because the people use these reservoirs for recreation. In this reservoir, during the weekends, we have 10, 20, 12,000 people. And they went to this reservoir because the water was safe and good water quality. No more. So there is also an economic problem for the region considering this infestation of the cyanobacteria. And you can see here, this was taken by my grandson. He manages to use these drones, and I say to him, take a picture, and then he took a picture of you could see the chlorophyll and the houses around the reservoir. So there are economic losses that will be surely associated with this infestation. And you could see the amount of chlorophyll. The amount of chlorophyll was extremely high. We had 100 micrograms per liter of this single species of cyanobacteria. The World Health Organization, the limits of the World Health Organization are 20 micrograms per liter. to be safe, and we had 100. So in all these occasions, and we had this problem of growth very fast of the cyanobacteria. The other problem that we had, of course, associated with that, very high oxygen concentration during certain periods of the day. I don't know if you can see probably here. And of course, almost zero oxygen concentration during the night due to consumption by the respiration. So the classical oxygen fluctuation during a very heavy bloom of cyanobacteria in a eutrophic lake. And this is our pictures of this species. Associated with it came also microcystis, but the main species is this Cylindrospermopsis, is a colonial species. And when we did measurements about 2 weeks ago, you can see here, this was also done with a fluoroprobe. You can see here the predominance of the cyanobacteria. The blue-green are here. And all the other groups are much less represented. So what happens in this reservoir? For the functioning of the reservoir, we had low retention time. Floodgates were always open. The winds were a factor of making the lake polymitic, circulation, permanent circulation. And with the change that we observed and measured, we have low rainfall, floodgates closed. Still, we had winds in the same direction, but the retention time from less than 20 days went to 60 days. These were the causes. With the same phosphorus concentration, we have a wastewater treatment plant of a small city here in one of the tributaries. We have non-point pollution from the houses and the beaches in this area. But the main cause then was higher temperature, higher retention time, low rainfall. No more silica from the rain to stimulate growth of diatoms. And of course, we are looking at the services, ecological services of the reservoir, and now we are looking at the effects of this infestation of cyanobacteria in all these services, the ecosystem services that the reservoir provides. This is the plan that we had for the reservoir, but what I would like really to discuss now, to finalize, I have this paper here. If you are interested, I can distribute. I have given to some of you. It's a note that we did to call attention to this problem as one example of a global change and a consequence of a global change. And I also suggest you the reading of this paper by Pearl and Valerie Paul. Is a very good review of the links to global expansion of harmful cyanobacteria that was published in Water Research in 2012. And these two authors also, they did a very interesting summary of all the problems linked with the global change and growth of cyanobacteria, calling attention especially to the problems of load of phosphorus from non-treated wastewater, increase of water temperature, and the low flow of the reservoir due to less volume of water. And you can see here some pictures of freshwater systems impacted by cyanobacteria. Another paper by Pearl and Usman is very clear. Blooms like it hot. This paper was published in Science in April 2008, showing the connections between higher temperature, global changes, and the infestation of cyanobacteria in fresh waters. And you can see here experiments that show the Microcystis aeruginosa, that is one of the main species that are also spreading very fast, can grow faster in higher temperatures than other species. So the cyanobacteria blooms linked to climatic changes are due probably to the following processes. First, hydrologic changes. Second, the nutrient inputs and the availability of nutrients. In many countries, only 20–30% of the wastewater is treated. This is a stimulus for the growth of cyanobacteria. The increase of water residence time is another factor that is connected. When you have less water, you have to increase the retention time because you cannot withdraw water for multiple uses, and this is a factor that stimulates the growth. Water mixing conditions also change, because if you increase the temperature, you increase the possibility of more stratification in the reservoir and with a higher surface temperature. So there is a synergic activity of climatic change with the anthropogenic nutrient enrichment. Many reservoirs in Brazil, drinking water reservoirs, are in this condition. We were surprised, we were taken by surprise in this reservoir that we studied for 45 years, and suddenly we had this large bloom in the reservoir that produced a tremendous change in the environment. I'm studying the concept of the tipping point. And all these reservoirs, they are very near the change in functioning. And if you have only one factor that makes this system to change, to go to another step of functioning, they grow from one step to another step. So our reservoir clearly overpassed the tipping point and presented these results. But this is an example. San Francisco River, for example, is full of Cylindrospermopsis, all the reservoirs. All the reservoirs of the Tietê River in São Paulo, they have blooms of cyanobacteria connected with less rainfall, less volume of water, high retention time, and high nutrient input. I would like to call attention that if we look at water quality problems, you can see a progressive degradation of water quality from the mid-19th century until the present. And in this progressive degradation, you can see that the last step is the synergistic effect of climatic changes and climate variability and the hydrological changes with these persistent organic pollutants, heavy metals, and so on. So I think this is the big question for the next 50 years in the 21st century, how to deal with the water quality problems and how to cope with the climate change that are affecting the freshwater ecosystems of the whole planet. Thank you very much.